Okay, thank you. I hope you're doing great. I'm also doing great. My name is Sostim from the Funzo Academy. So, we are going to look at the photosynthesis, right? Particularly, we're going to look at the investigation. So, our investigation will be based on two aims. So, the first aim is to determine if a factor is required for the photosynthesis. We know that our photosynthesis it need a light, it need carbon dioxide, is also need a chlorophyll. Already have been told that, right? So we are not sure whether what we're told is true or is not true. So we're going to do the experiment to determine if indeed light is required for the photosynthesis, if CO2 is required for the photosynthesis, if chlorophyll is required for the what? For the photosynthesis. Remember, this chlorophyll, it means is the what? It is the green pigment. It's the one that gives the leaves a green color. So we're going to determine if it is required for photosynthesis or if it is not required for the what? For the photosynthesis. So also we're going to determine the rate at which photosynthesis is occurring. So the word rate here, I underlined it. It means the speed. We're going to determine if photosynthesis will occur very fast or it will occur very slow. If the light is increased, will photosynthesis occur fast or will occur very slow? If the carbon dioxide is increased or decreased, will photosynthesis occur slow or very fast? So during our investigation, we have two parts. So we have a part, we call it a what? We call it a control. And we have the parts, we call it a what? We call it an experiment. So what we mean by the control? Control, it is the plants that we are going to give all the requirement that are required for the photosynthesis. So we're going to give it CO2, we're going to give it a light, we're also going to give it what? The chlorophyll. So the control, it have the what? They are all the factor that's required in the photosynthesis. But experiment part, it will have all other factor except only factor that we are investigating. For example, if in our experiment we're investigating whether light is required for photosynthesis, it means we'll have all other factor. We'll have CO2, we'll have chlorophyll, except the what? Except the light. At this part, we don't have the what? We don't have the light. There's absence of the light. That's what we call a what? We call it a what? Experiment. We call it experiment. Experiment, there is no what? There is no factor that is required. It is what? It is absent. It is an experiment. Control, we have all factor. And the importance of the control is what? It's important so that we compare our what? We compare our results with the what? With the experiment. It's very important so that we compare the results with the what? With the experiment. That's the importance of the what? Of the control. Or we can say it's act as a what? As a baseline. After photosynthesis, we know there is a formation of the what? There's formation of the glucose. And this glucose will be now converted to the starch. And this starch should later be stored in the what? In the granules. So all our experiment will be based on the what? Will be based on the starch test. So if the starch is present, if we perform a starch test and we find that the starch is present, it means now the photosynthesis is what? It is occurring or it has occurred. But if the starch is not present, it means there's no what? There's no photosynthesis. For example, if there is an absence of the light and we find out that there is no starch, it means photosynthesis did not take place and the light is required for the photosynthesis. If there is no light now, there's no chlorophyll, and we find out that the starch was formed even when there's no chlorophyll, it means now photosynthesis can take place even without the what? The chlorophyll, because the starch was what? The starch was formed. In our investigation of photosynthesis, we must make sure that we perform this process called a what? Called distarching. So what do we mean by the word distarching? The word distarching means removal of the what? Of the starch. Before we perform our investigation, we must make sure that we remove all the starch that was formed earlier before our investigation. So we know in environment, photosynthesis always occur in what? In a plant, because there's all other factor. There's a light in the environment, there's there's a what? There's a carbon dioxide that's always released in the environment. There are always a what? There's always a chlorophyll in a plant. So all the factors are there, and it means photosynthesis will take place, and there will be a what? There will be a starch. So before there will be a what? A formation of the starch that will form earlier before our experiment. So we must make sure that we remove all the starch that were formed before experiment, so that it does not interfere with our what? With our results. So the starch that we will be testing now, it will be the starch that were formed new or due to our investigation, which is the new starch. We remove the old starch, and now we're testing the new starch that were formed due to our investigation, not due to the environmental factors. So how do we remove the starch? We remove the starch by doing what? By placing our plants in a dark cupboard for the what? For the 48 hours. And during these 48 hours, all the starch that was stored will be what? Will be removed. Will be used by the what? By the plants. So that's the aim of the what? Of the discussion. I think now we're done. Gonna see you at the what? At the next video.